All right. So yeah, I think that we have over 20 people here. So it's my great pleasure to introduce our today's speaker, Dr. Hassan Samee. Hassan did his PhD in University of Illinois at Urbana-Champaign in Department of Computer Science. Actually, Hassan and I were from the same lab and overlapped for a couple years. And we had a publication together because we shared our PIs. We were among the first, I guess, PhD students of SORAP in UIUC. So that's good. I guess like you came a couple years after me. Hassan was extremely successful during his PhD. He went for another successful postdoc experience in University of San Francisco at Gladstone Institute. He did an amazing job during his postdoc, had multiple publications, and he recently became an assistant professor, I guess in January 2019, in Baylor College of Medicine. And since then, he has been also publishing several works. I have seen some of the bioRxiv works that looks extremely exciting. So he has been absolutely fascinated with statistical models and machine learning techniques and to study different aspects of biology. And Hassan is going to tell us a little bit about his recent work. And I'm not going to take more time from you, Hassan. So please go ahead.
Thank you so much, Majid. That was a really kind introduction. And before even I start, I would like to say that Majid has always led us with examples. So thanks, Majid, for leading us with examples. So this is a really, really great privilege for me to speak in this seminar. So I'm presenting our work today on protein DNA binding to spatial transcriptomics for studying transcriptional regulation at multiple scales. So this is the phenomenon that our lab is mostly excited about, gene transcription, which means production of messenger RNA, mRNAs, from genes, the protein coding sequences of our genome. And of course, this is one of the levels of biological functions, starting from mRNA. The mRNAs gets translated into proteins and so on. We are primarily focused at this level of gene transcription. And proper level of gene transcription, meaning producing the proper amount of messenger RNAs, is important in physiology, and disruption in those may lead to diseases, as we will see in some examples coming down the slides. So if the proper regulation of gene transcription is disrupted, it may get down-regulated, meaning that we start getting fewer mRNAs from a gene than is necessary to maintain physiological conditions. It may get upregulated, meaning that we may start getting more mRNAs than necessary. So the two things that fascinates us as computational scientists about gene transcription, first is it is a very systematic process. What do I mean by systematic? So in this plot, for example, which I took from the Human Proteome Project, what we are showing here is the transcription level of 3 different genes in different organs. So the y-axis here is transcript per million, which is a unit to tell us how much messenger RNAs, mRNAs are being produced. So for POU3F2, as we see that it is expressed only in cerebral cortex, not in heart or spleen, NKX 2.5 in heart, and about half as much in spleen, but not at all in cerebral cortex. And EEF1A1, it's again very high in spleen and about half as much in spleen in the other 2 organs. And it's not only this kind of high, low, and medium level. If we go further, we can see that proper transcription of EF1A1 means like systematic levels across the entire body. And changes in these transcription levels are often associated with diseases. So some known and very visual examples come from osteoarthritis, for example, where downregulation, meaning that less messenger RNAs of the gene GDF5 being produced, can lead to this kind of thickening of bone joints, osteoarthritis. On the other hand, upregulation of the sonic hedgehog gene, SHH, may lead to polydactyly, as you can see, like additional limbs. So these are the two main points that drive us as computational people to study these biological phenomena. Again, A, this is a very systematically, quantitatively maintained process. And B, the changes in transcription levels are often detrimental. So that takes us to ask, where are the information encoded for gene transcriptional regulation? And this has come from decades of research that the non-coding parts of our genome, meaning that the parts that are outside of the protein coding sequences of our genome, those non-coding DNA encode the information to regulate gene transcription. So there are specific regions in the non-coding genome called enhancer, and these are like kind of hidden in the about 97% of our non-coding genome. And these enhancer sequences, they are associated with the genes. The enhancer sequences may be distal up to megabases. And these enhancer sequences, they encode binding sites, as we are showing here, binding sites for regulatory transcription factors, where the transcription factors bind. And upon binding, they regulate gene transcription. And in fact, the examples that we saw in the previous slides, they are classical examples of gene dysregulation because of disruption in transcription factor binding. So the osteoarthritis example, where we saw that GDF5 is downregulated, it gets downregulated because in the enhancer sequence associated with GDF5, there is a mutation that disrupts the binding of a protein called YY1. And likewise, the upregulation of the sonic hedgehog gene associated with polydactyly, that happens because of the disruption of ETV, that transcription factor is binding to its enhancer sequence because of a mutation. So that actually takes us to study transcriptional regulation at multiple scales. In one scale, we want to understand how do transcription factors recognize their target binding sites. And in the second scale, we want to understand how do DNA-bound transcription factors regulate gene expression. So in my presentation today, I will try to share some of the things that our lab has been trying to contribute toward these directions. On the first point, I will try to describe the role of DNA shape, DNA structure in transcription factor DNA binding specificity. And on the second point, I will describe a model where we are jointly learning the transcription factor DNA binding specificity and transcriptional regulation in data sets coming from massively parallel reporter assays. So these are more at the scales within nucleus, but more recently with our collaborations in Baylor College of Medicine, we are also studying transcription and regulation at the cell-to-cell communication level. So we are leveraging the power of single-cell transcriptomics and single-cell spatial transcriptomics to study how does a cell's spatial location influences transcriptional regulation in it. So I will come back to this visual later, but for now, this is what we mean by single-cell spatial transcriptomics. We have slides. I believe that this audience already knows this, so pardon me if this is like unnecessary. So these are spatial transcriptomic slides. Each slide has tiny spots or beads, and we put a tissue section on these beads, permeabilize the tissue section. So the cells, they open up, enter into the beads, and then we do single cell transcriptomic within each bead. So finally, it gives us this kind of a data matrix where each row is a cell, and we get the gene expression values of each cell, just like as in a single cell RNA sequencing data, but we also get the XY coordinate of that cell in that tissue slice. And we are trying to leverage this powerful data modality to understand a cell's transcriptional regulation happening within a cell, taking its spatial location into consideration. And what we will very briefly try to describe is the spatial organization of cells and intercellular communication. Those are two major things influencing transcriptional regulation inside the cell. So I'll start my first part of the talk about the role of DNA shape in transcription factor DNA binding specificity. And we all know classically that transcription factor DNA binding is understood as a sequence specific mechanism. What do I mean by that? So most often we know that when a transcription factor shown as this red ball here binds the DNA at a binding site, which I'm showing here as an example, ACA-GTCA, it means that most often this transcription factor will bind to minor changes of the same sequence. And we generally model this using this kind of a visualization, which is actually the visualization of a multinomial model, which we also call sequence motif, which tells us that wherever the transcription factor binds, if you take one of those binding sites, with high probability, we'll see that there is an A in the first position. And almost always we'll see there is a C in the second position. It's okay to have a G. It's also okay. It's mostly preferred. I should say it's mostly preferred to have a G. It's also okay to have a C at the fourth position, and so on. So that's how we have been modeling transcription factor DNA binding as a sequence specific mechanism. However, we also know that sequence preferences cannot explain transcription factor binding in many regions genome-wide. This same transcription factor, maybe we will find another place of the genome where it's bound to a location where the sequence is completely different, like GCTA, TGA. So we started asking, and it's actually something that started around 2012, 2013, when Dr. Remo Rose in University of Southern California, he started bringing up the role of DNA shape in transcription factor DNA binding at a genome scale. So we started asking that, okay, if Dr. Remo Rose is looking at the shape of transcription factor binding sites based on the sequence motifs, can we go one step further? Is it possible that a transcription factor, it can recognize two different DNA sequences, even though they are completely different in terms of nucleotides, but they could be very similar in terms of their DNA shape properties? So for example, here, these two sequences, if I look at the role values, so role is the angular measurement that I'm showing here between two neighboring dinucleotides. So is it possible that this is what's happening in some cases of a protein DNA binding? So the role profiles of these two sequences are remarkably similar. So we wanted to pursue this hypothesis that is it possible that a transcription factor can recognize its DNA binding sites, not only based on nucleotide sequence similarity, but also based on DNA shape similarity. So before I go to discuss the whole idea, I would like to spend just one slide to bring everyone on the same page that what do I mean by DNA shape in general. So DNA shape profiles, they actually represent the geometric features of DNA at nucleotide resolution. So again, we have been looking at DNA sequences as linear nucleotide sequences, but in reality, DNA is a three-dimensional molecule. In this left panel, I'm showing a cartoon of the DNA molecule. The yellow ribbons, they are the backbone, and we have the base pairs here. So if blue is A, red is T, if yellow is C, green is G. And what we can appreciate by looking at this left panel is there are linear and angular displacements between these nucleotides and between neighboring dinucleotides, between neighboring base pairs. So These angular measurements, they create the DNA shape profiles representing shape features at nucleotide resolution. So these angular and linear displacements between bases and neighboring base pairs, they define the DNA geometry. And more anecdotally or classically, there are 4 shape features that have been investigated very well, and they are known to play some role to influence transcription factor DNA binding. These 4 features are helical twist, minor groove width, propeller twist, and roll. So we just described roll. If we look at propeller twist, it means how twisted are 2 nucleotides, are 2 bases in the same base pair. Minor groove width is more classical. It is the opening between the bases in the same base pair, and the helical twist, it's again between 2 neighboring base pairs. And we started asking this question, focusing on these 4 more classically studied DNA shaped features. So going back to our previous example, then we said that, okay, we are familiar with this sequence motifs and sequence specific mechanism. Let's investigate into the existence of shape motif and probably a shape specific mechanism. And what we found from our analysis, so we analyzed all the ENCODE data sets, which is like hundreds of transcription factors covering about 10% of the human genome. We used all known sequence motif finders and the curated libraries of motif hits based on sequence motifs. And we found that about 30% of the CHIP peaks, the experimentally assayed locations of transcription factor binding, actually lack any sequence motif, any known sequence motif for these transcription factors. So then we started building a model to test this hypothesis that transcription factors, they recognize shape patterns. And here I'm very briefly explaining the model itself. It's called Shape MF, Shape Motif Finder. Essentially, this is a classification model. We take positive sequences, meaning examples of the transcription factors bound locations, and negative sequences where the transcription factor does not bind. We translate the sequences into their shape profiles. And once we translate them, they look completely noisy, right? Because even from DNA sequence-based models, we know that in a given region of a transcription factor's binding, the exact location of the binding site may happen anywhere within these 50 or 100 base pair regions. So then we take a more classical approach, a Gibbs sampling based approach, to select one window, one small stretch of these noisy signals per shape profile from positive training sequences. And for Gibbs sampling, again, we use a very simple energy function based on the Euclidean distances, which pulls out this kind of short and very similar looking signals from the input positive sequences. We haven't used the negative sequences yet in this process. So once Gibbs sampling pulls out these very similar looking signals from the positive sequences, we see that there are some sequences for which the shape profiles do not match with the rest of the sequences. So then,
can I interrupt you just for clarification?
Yes.
How does this shape profile created in general? Maybe I missed that.
No, that's a very good question. Yeah, sorry, I should have mentioned. So Majid's question is about this step where we start with a set of DNA sequences, and I said that we translate them into DNA shape profiles, and the translation comes from data from the Remo Rose lab at University of Southern California. So what the Remo Rose lab did was they simulated thousands of DNA sequences. Each sequence was about 40 base pair long. So they simulated the DNA sequences through molecular dynamic simulation until the DNA sequences reached equilibrium. And at equilibrium, they measured the angles for all of those sequences. And then they built a model for those sequence to DNA shape. And they found that it's a 5th order Markov model that captures the shapes the best. So then they created these tables of pentamers of 5 nucleotide long DNA sequences. And now for any arbitrary DNA sequence, we read the sequence in 5 nucleotide long stretches and just take the values from those tables as the shape translation for the input nucleotides. Thanks for asking that question, Majid. I hope that's
clear. Thank you.
okay. So so then we see that there are some shape profiles which are not highly similar to the other ones. So at that point, we ask that, okay, we will build a shape model taking the negative sequences, the DNA shape profiles of the negative sequences into consideration. We will say that each value, at each position, the DNA shape values, we just assume, but we saw that the assumption holds quite well. At each position, the DNA shape values, they follow a Gaussian distribution. The mean, we take it from these values. But for the sigma, the standard deviation, we gradually increase the sigma to an extent where we see that we are not able to discriminate between the positive and the negative sequences. So basically, we say that this is optimizing the multiplier for sigma values until we are significantly well discriminating between positive and the negative sequences. And once we have done that, we come out with this kind of a DNA shape pattern where the dark values, they tell us the average at each position for one particular DNA shape feature. It can be helical twist or propeller twist. And the shaded regions outside the dark value tells us like how much tolerance do we have at that position for the DNA shape feature. And these descriptions, if you call these like multidimensional Gaussians, they are our shape motifs. And we define them as the shape patterns that have a significant p-value in discriminating between separately held out positive and negative sequences. So we optimize these K, the multiplier of sigma on the training data, and we finally validate them on the held out test data. And we report only those shape patterns that are significant in the test data. So that's our shape motif construction. And we found that in the ENCODE data, more than 80% of the transcription factors, they have shape motifs. We also validated those, not experimental, I shouldn't use the word validated. We also checked for the existence of shape motifs in vitro data, which are more quote unquote purified proteins tested in vitro HD-CLEX experiments. And we discovered very similar shape motifs. So for example, here for PAX5, the green things, the green plots, they are the chip signal derived motif. The purple plots, they are the CLEX derived motifs. And we see that they are very similar for all these examples. And in general, I said 2 slides ago that about 30% peaks of a ChIP-seq data set lack any sequence motif, but we find that in general, 20% of the ChIP-seq data sets, they lack any sequence motif, but they do have a shape motif. So there are about, on average, still 10% peaks of ChIP-seq data set that we cannot explain yet. The point that excites us are about 60% occurrences of shape motifs that do not overlap with sequence motif occurrences. So the 30% chip peaks where we did not find a sequence motif, those were not the quote unquote weak chip peaks. Those were equally distributed in the strongest chip peaks and throughout the spectrum of chip signal. And similarly, we saw that even in the strongest chip peaks, we find shape motifs occurring alone or shape motifs occurring with a systematic distance from the sequence motif. This paper was published in Cell Systems early 2019. I will just touch upon one interesting part of that paper and move on, which is we saw that transcription factors within the same family, they recognize very distinct shape motifs, and which kind of answers one of the questions that if different transcription factors of the same family have very similar sequence motifs and are expressed in the same condition, how do they recognize different locations in the genome? So here I have one example for JUNB and FOS, and the shape motifs that we are showing here are helical twist motifs for JUNB and FOS. And the known sequence motifs for these two transcription factors are remarkably similar: TGA with a less specific nucleotide in the middle, followed by TCA. And as we can see that the shape motifs for these two transcription factors are remarkably different. The length are still very similar, but there is a sharp dip toward the end for June B, and for FOSS, it stays at around like 34 to 35 angstroms. So again, the paper was published. This is only one part of the exciting story, at least the story that excited us. I would welcome more criticisms and comments from this audience, because we are still trying to pursue these ideas of DNA shape. And I do not have everything today, but we recently submitted another manuscript. The bioRxiv version is there, where we have found that DNA shape, it actually correlates very well with mutation rate variations in the human genome. So we took DNA shape to study polymorphisms in the 1,000 genome data. And we actually saw that this is a very strong correlation in the entire non-coding genome. We came back to transcription factor binding sites. We asked at each position of transcription factor binding sites, how does DNA shape variations correlate with polymorphism in position-specific mutation rates in transcription factor binding sites? And for all the transcription factors, we found that there is a strong negative correlation, meaning that the bigger is the deviation in DNA shape, the less likely is that particular position in transcription factor to be polymorphic in the human genome. So we are excited, and there is another project that I'm continuing with my postdoc advisor, where we are studying the evolution of DNA shape. And again, we are finding interesting things. So when we started this project, there were many questions coming to us, both from enthusiastic angles and both from highly critical angles. And one of the highly critical points was, is it just artifacts of data processing or the statistical models? And well, we haven't yet found a very strong way to test these hypotheses, but what we are seeing is very similar and intuitively coupled ideas are holding across different data sets, starting from transcription factor DNA binding to human polymorphism and cross-species evolutionary data comparisons. So I will move on to the second part of my talk here, which is joint modeling of transcription factor DNA binding specificity and transcriptional regulation in massively parallel reporter assays. And these works, they build on a probabilistic graphical modeling approach of modeling gene transcription, which was initiated by Shin Hee, Dr. Saurav Sinha's, I think he's first PhD student of Dr. Saurav Sinha, now a very successful faculty at UChicago. I was proud to have my first paper with Shin in 2010. The basic idea of these models are, we have these enhancer sequences associated with the genes, and we also use the known transcription factor DNA binding motifs to mark the transcription factor binding locations in the enhancers. So then we can say, in a thermodynamic formalism put forward by Shia and Akers in 1985, that this whole system can stay in one of these exponential number of possible configurations. So BTM here is the basal transcription machinery, which is kind of a lump variable combining all other necessary machinery for transcribing this gene. So now Shia and Akers suggested that we can model this formalism under this thermodynamic framework, where one configuration says that there is no transcription factor bound, but only the BTM is there. Another configuration says that there is only the first transcription factor with the BTM, and so on. So Shia and Akers suggested that we can model the observed gene expression data as the expected value of gene transcription coming from these exponential number of configurations. The probability of each configuration with combined or multiplied with the expected, with the gene expression coming from that particular configuration. And what Shin showed was this whole formalism can be modeled very efficiently as a probabilistic graphical model through a dynamic programming formulation. And in my PhD, we took this model forward to modeling a couple other directions, which I have cited here. But one of the key things of these models is we need to know the motifs a priori. So we need to know which transcription factors we are considering, and we need to take those motifs and scan the enhancers to mark the locations of those motifs. So this is quote unquote a limitation that we wanted to overcome. And now we have more data coming from massively parallel reported assays. So we wanted to overcome this limitation, taking the power of convolutional neural networks. And this is not exactly a new direction per se, because convolutional neural networks, they have been used highly in genomics, starting from the paper called DeepBind by Babak Ali Panahi and DeepC from Olga Transka's lab. We have seen many successful CNN convolutional neural network models in genomics. However, all those models need post-hoc analysis to identify sequence-specific signals. In other words, to identify which parts of the input sequences really matter to the model. And some of those post-hoc analysis approaches, such as deep lift from Anshul Kundaje's lab, they indeed come back with something that is a motif, that is a sequence pattern. But those models, those post-hoc analysis models, they make additional assumptions about linearity of the whole deep neural network, which does not guarantee that if we take the same motifs and plug them back into the initial model, shall we get an equally good model? So, and also those motifs coming from models like deep lift, those motifs, we don't know for sure if we can use them as a general purpose motif that we have found from other biochemical studies, because they are not founded in a biophysical formalism. So what we thought that we could simply try a tweak in how we apply the convolution operation. So the classical convolution operations is essentially an element-wise dot product. We said that can we use the convolution operation to perform a likelihood ratio test. So for example, we said that if X is our convolution, we will transform each X, each convolution matrix, into a multinomial distribution. This is a simple Boltzmann distribution transformation. So we take each X, exponentiate it, and divide it by a normalizing factor, which is the summation of the xi values for the xij values for all 4 nucleotides. So by doing that, we easily transform each convolution into a multinomial distribution. And then if we have a genome-wide background distribution also, then we, instead of taking an element-wise dot product, we are actually computing a likelihood term as a result of the convolution operation. And once we do that, we actually get the likelihood for a motif match across the entire input sequence. And then it becomes a simple linear regression. The type of model that Eric Sigia and Herman Bussemaker, Hao Li, they had published in 2001 in Nature. Also Majid and Charles in their PhD work had published in PLOS Biology in 2010. So it starts with a very biologically grounded sequence motif match in the input sequence. And then we can do many things, more mechanistic, less mechanistic. But the point is, once we have these multinomial distributions, they're completely biophysically interpretable. And we can take these motifs even outside the context of this model and use them in any other way that we have used motifs from the classical motif databases. So this was, again, I won't say that this was a groundbreaking contribution, but this was more of testing an idea. What we found encouraging was we started finding models that had much fewer parameters than other convolutional networks tested on massively parallel reported datasets. And yet we improved over state-of-the-art performance. We discovered the relevant transcription factor binding sites from a liver enhancer MPRA data, and we did not even need to take any epigenetic information into account. So the previous best models, they needed to take many epigenetic information into account. We were able to outperform those models simply based on these more biophysically grounded sequence motifs. And we also tried to find more applications of these models. So we fit the models on MPRA data, did not retrain the model further, but then we applied the model to predict genome-wide accessibility in the same cell line. And what we are showing here is our model captures very well the rank of the genome-wide accessible sequences based on DNA-seq data. So this is what our model predicts: the activity of 150 base pair location in the genome. And this is how the sequences are ranked according to DNA-seq data, and they're very highly correlated. And again, we did not retrain the model. This is the model coming from the MPRA data. For benchmarking purposes, we were having an exciting discussion before this talk about reviewer comments. So the reviewers have asked us to do more benchmarking. For benchmarking purposes, when we are retraining the model on DNA-seq data, we are actually doing even better. So the whole purpose of our manuscript was to show that this new way of fitting the models and learning convolutions as multinomial distributions give us a very good, easy to interpret model in terms of the convolutions. We can avoid all the post hoc analysis approaches, yet we do not lose any performance. And that's what we saw. And now in benchmarking, we actually are seeing that even stronger. We also applied the model to predict the effect of potentially, to identify potentially functional SNPs in the human genome. So very briefly, we took all the common variants from DB SNP. By common, we use here a cutoff of 1% minor allele frequency. And we asked to our model that for each SNP, what is the predicted change in model predicted activity from the reference allele to the minor allele. And then what we find is the most significant changes predicted by the model are enriched for the lowest minor allele frequencies, which goes with the hypothesis that if a sequence's activity change is highly remarkable, it is probably avoided in the human genome in the polymorphism data sets. So again, these are demonstrations, if you will, demonstrations that the model is showing us good results without sacrificing performance. And based on reviewers' comments, we are doing more benchmarking on this aspect of the model as well. But the two ideas that actually have helped us a lot to move forward are A, these sequence motifs are highly usable in other contexts outside of the model itself. We could very easily interpret the model. And once we have these log likelihood ratio computations on the input sequences, we could readily move to building more mechanistic models, more mechanistic than a simple cascade of convolution and pooling can do for us. And secondly, we are now able to avoid the GIP sampling approach in our shape motif finding, because here we are actually saying that the convolution operation can be treated as an operation that tests for the likelihood of a generative model generating the input. So the multinomial distributions here are the generative models. And for the DNA shape motifs, now we are learning the convolutions as multivariate Gaussians. So we have just a vector of 2 columns, and let's say for a 10 nucleotide long binding site of 10 rows. So each column is either so column 1 is the mean, column 2 is the variance, and we are learning the mean and the variance parameters in those Gaussian convolutions. So each convolution operation now tells us that what is the likelihood for the observed DNA shape to come from this multivariate Gaussian. So we are directly learning those multivariate Gaussians as the convolutions of our model. And then we are visualizing those multivariate Gaussians as our shape motifs.
I am sorry, Hassan, can I interrupt you just for clarification?
Yes, yes, sure.
Um, so this convolution neural networks that's transparent and you trained, is it on the shape data or was it on the massively parallel reporter data? And I'm a bit confused on which of those.
Oh, sorry about that confusion. So whatever I said so far is trained on massively parallel reporter assays. But then I was just sharing some excitement that we are now taking the same approach of learning a probability distribution to our shape motif discovery pipeline. That actually we have now realized that shape motifs, so when we are applying Gibbs sampling, we are getting fine results, but still the models face some scalability issue. But in this approach, we can learn each convolution as a multivariate Gaussian. And the rest of the things stay the same, and we are learning equally effective models. So yeah, thanks for asking, Majid. Sorry about the confusion. So this is where I will switch gears to the last part of my talk, where we are taking advantage of single cell and spatial transcriptomics data to learn a little bit more on transcriptional regulation. And what I'm going to show you is our analysis suggests that spatial organization of cell and intercellular communication between cells influence transcriptional regulation. This third work, it's also available in bioRxiv. We were able to excite the reviewers. The bad thing is they have asked us for validation. But for both like the second work and the third work, please share your criticisms and comments with us. And if you have any other data set in mind, we would love to apply those models on these data on the data sets from your suggestion. So this is kind of the background slide where we are taking these two data modalities, where we are combining these two data modalities. The point is combining these two data modalities come with some challenges because single cell transcriptomics and spatial transcriptomics, they bring complementary information. So here is a mouse olfactory bulb. This is the subject of our bioRxiv study. So this is a mouse olfactory bulb. If we take a slice of the bulb, it looks like this kind of a tissue slice. And the data set that we have studied actually picks up 6.2 by 0.2 centimeter, so 2 millimeter by 2 millimeter regions from these tissue section. So each of these sections is called a field of view, FOV, and gives us the gene expression value of each cell with their XY coordinate from each of these field of view, each of these FOVs. The interesting thing is in spatial transcriptomics, we get the XY coordinates. So the data table in spatial transcriptomics looks like this, that we have each cell and we have the value of a bunch of genes with the XY coordinate of the cell. To compare this with single cell, like the regular single cell RNA-seq data matrix, again, we have the cells and the expression values of a bunch of genes. We lose the XY coordinate in single cell data processing. So apparently spatial transcriptomics should be more powerful, but it turns out that spatial transcriptomics as of now profiles fewer genes. So single cell RNA-seq profiles many more genes, almost like more by twofold. So that actually limits our ability to assign cell types in spatial transcriptomics data, because to assign cell types, we need marker genes. I believe this audience doesn't need that introduction. So we need marker genes. And in spatial transcriptomics data, we may actually find only 1/3 of the marker genes that are necessary to confidently assign cell types. So more visually, it looks like this. So we had a mouse olfactory bulb single cell RNA-seq from our collaborators. The data was very well separated into different cell types. And for the same mouse olfactory bulb, when we take spatial transcriptomics data coming from a technology called seq-fish+, it's a fish-based technology, we have the XY coordinates, but we do not have the cell types. So we built a very simple multi-layer perceptron fully connected neural network model to learn to assign cell types to the cells in spatial transcriptomics data, having input as the genes that are shared between single cell RNA-seq and spatial transcriptomics. So the model can tell us that, hey, these cells, they belong to the blue category, these cells, they belong to the red category, and so on. So using this simple multi-layer perceptron neural network, we were able to transfer the cell types to each of these cells in the spatial transcriptomics data. And then we started asking some interesting questions about cell-cell co-localization, for example. So what we found here in our first analysis, we took two FOVs. So here I'm showing FOV2 and FOV3, which are both coming from the innermost part of mouse olfactory bulb. And what we can see here is in these two FOVs, although they're in the same architectural layer of mouse olfactory bulb, there are cell types who show very different patterns of co-localization. So for neuron transition cells and olfactory and sheathing cells here, we see that in this FOV, which is FOV2, they do not co-localize to an extent that they co-localize in FOV3. So if we look at this like more crisp figure, we can see that neuron transition cells and olfactory and sheathing cells. So these are the olfactory and sheathing cells, and these are the neuron transition cells. They have very few areas of like strong co-localization in FOV2. As compared to FOV3, they're like completely co-localized. So this was our first interesting observation in this data set, that we are able to see architectural principles beyond the layer-based description of brain. And actually that is the like key point that we have discussed in our bioarchive paper. But this whole idea of co-localization also took us to ask, well, if there is a change in co-localization, does it also imply changes in transcriptional mechanisms, if you will? So we asked, if two cell types, they show different co-localization, how different they become in cell-to-cell communication mechanisms. So we went back to some published computational models to infer cell-to-cell communication mechanisms. And for every pair of cell type, we asked that what is the minimum number of receptor and ligands that they use in one FOV, and what is the maximum number of receptor and ligands that they use in a different FOV. And we saw that there is a huge variation in the usage of receptors and ligands. On average, it's like 3 to 4-fold change in the use of receptors and ligands between the same two cell types from one FOV to another FOV. And it could go as much as around 19-fold change in the use of receptors and ligands. So that was, again, telling us that there are big changes in cell-to-cell communication mechanisms through expressing receptors and ligands. So then the question is, can we look at the gene regulatory networks connected with these receptors and ligands in these FOVs? And is it the case that the receptors and ligands in the different FOVs, they're completely connected with very different sets of genes? So for each receptor and each ligand, we actually constructed the gene regulatory networks in the different FOVs, and we asked how many genes are actually shared between two FOVs. And as you can see that there are actually very few, on average, only about 20% of the genes are shared for the same receptors or the same ligands gene regulatory network across the FOV. So which tells us that there are big changes happening based on the spatial location of the different cells, and potentially that also regulates how the cells are communicating with each other. Now, this is just one data set, and the spatial transcriptomic technologies are also becoming more enhanced in terms of the data quality. I think everyone knows that. But we already have seen that some of these observations have interesting implications in the tumor microenvironment in terms of immune cells and the stromal cell communication. So we are actively pursuing those directions while addressing the reviewers' comments. So with that, I would like to conclude my presentation here today. Before I conclude, I would express my gratitude and acknowledgement to my postdoc advisors and co-advisor, Katie Pollard and Benoit Brenu, because they allowed me to pursue the shape motif idea, which was at that point, again, many renowned names criticized us, but also we got huge support from some very remarkable names, Peter von Hippel and Ron Shamir. I would always stay grateful to them. The lab, we are a fairly new lab, and of course the COVID thing have stricken us in terms of our recruitment and so on, but each of the lab members, they're remarkably productive. Xian Liu, he is actually a Purdue undergraduate, so he has brought a lot of energy to the lab. Xian actually produces more plots that I can process in a day. So he is very energetic. And Xian is actually now leading the effort on modeling mutation rate variations using DNA structure. We have been funded through a bunch of funding sources here. And I'm also very grateful to my collaborators. They're actually allowing us to do more computational things, not only data analysis. So Jim Martin, he's a famous name here on heart regeneration. Diwakar, he's a pediatric surgeon. With Diwakar and Jim Martin, we are also looking at pediatric heart samples. Susan, she is our department chair and also a very supportive mentor for me, I would say. And with Susan, we are looking at the epigenetics of metabolism, some of the data sets we didn't touch today. Robia, she is an Alzheimer's disease person, very famous name in the community. With Robia, we are looking at the spatial transcriptomics and the histology images. We are trying to connect those two coming from Alzheimer's disease mouse models. And Lynn Ziedrich, we are collaborating with Lynn on DNA structure. They have exciting hypothesis on DNA supercoiling, and together we are trying to connect how DNA structural changes might make certain regions of the genome more susceptible to mutations and potentially leading to diseases such as cancer. So finally, I would appreciate any and every comment and suggestion from this audience. So please feel free to email me at my last name at bcm.edu. And I would like to thank you for your time and take your questions.
Thank you so much, Hassan. Let's thank our speaker, although we don't have any kind of like in-person visit, hopefully next time. This is extremely exciting, and I didn't know that we could take the old dog and learn new tricks, and a lot of basically grating stories that are coming out of old motif finding that we thought like that's just a done thing, and then there's a whole new area of research that you are moving forward. So let's open up for question. I have a lot of them, but I'm just gonna let anyone who might have questions to go ahead and ask.
Yes, please.
Then can I start the asking questions? Actually, first I have a question in the shape. So our lab is working on more like a 3D shapes of structure modeling and so on. We haven't done much about DNA, but actually when I came to the Purdue, well, I was thinking to switch my area to DNA and RNA because my to avoid some overlap with my former advisor, who is a very well-known person in structure modeling. So at that time, I have seen those shape ideas. It has been already a long time ago. So okay, the question I have is that it's very interesting here. Actually, two things. One is that when you find the DNA sequence which have the same shapes, usually are bound by the same transcription factors, proteins. so when you align the DNA sequences with the same shape profile, does it mean that those DNA sequence is controlled by the same transcription factor?
So
Not necessary.
that's right. We have found examples that two completely different nucleotide sequences may
Right.
have a similar shape.
Right.
One point about these shape data are they are generated from quote unquote linear or naked DNA sequences, right? So they are simulated without any other molecule attached to them.
Right. Right.
So one question that we often get is the shape profiles that you are using, are they before transcription factor binding or after transcription factor binding? And again, most of these questions, they need more experimental validation. What we, and Remo Rose, the lab who actually started generating these data sets, what we think is these are the shape profiles are more inducive for the binding of certain transcription factors. Um, to go back to your question, it could be two completely different DNA sequences.
You mean completely different transcription factors may be binding there?
Exactly.
Oh, okay. So actually, okay, the reason I'm asking is that now you have some idea of the shapes from DNA side. And then like from protein side, I can see, because there was, well, some number of crystal structures between DNA and protein complexes. So, and then by using those structure information of proteins, we may be able to guess or predict model structure, 3D structures of transcription factors.
Totally, yeah.
Yeah, so by looking at the transcription factors DNA binding site, it should have a complementary shape to the DNA shape. So by looking at the interface DNA binding site, the shape of the DNA binding site of transcription factors, do you think we can predict binding sequence by shape matching?
I would say this is possible, and this actually opens up a lot of new questions, right? I was looking at this new paper from Barry Honig's lab, where they were talking about chimeric viral proteins. Like at the sequence level, the viral proteins, they do not look very similar to the human sequences, like the protein coding sequences. But in 3 dimensions, I guess you are an expert to think about that direction. But they found that many viral proteins, when they fold and become the full 3-dimensional protein, they become chimera of the human proteins. And when we reviewed the paper, we were thinking to ourselves that is it possible that now they also start competing for the same DNA binding sites and control regulation in translation or transcription of gene and so on. So I think this is totally exciting and possible to check if given the shape of the protein DNA binding interface, like the DNA binding domain and the shape.
Right. I know some people is trying to do a structure-based prediction of binding site. Well, if a transcript shape 3D structure of transcription factor is known, they thread DNA along the DNA binding site of that protein, and then compute the energy, binding energy at each position of the between protein transcription factor and DNA sequence, and then predict where they bind. It's like a good old magnetic tape going through the reader. You thread the DNA sequence along the transcription factors, and then you compute binding energy up and down, and then identify where it really binds in DNA sequence. So in that case, those guys are using statistics of the protein atom interaction against DNA atom interaction. It's a statistical potential, but maybe we can consider more like a coarse-grained shape that you described to see where they bind, and then we can do it probably massively because you can get the shape information from just from DNA sequence. It looks like it's working remarkably well. So then somehow add the structure information into this transcriptomics world. And I thought that could be wonderful if we could do that.
Totally. I mean, we are totally excited to move on to these directions because, again, I feel that there are huge untapped potential in in quote unquote structural genomics. Like we have spent enough time. I often say to my colleagues that quote from Eric Lander that DNA bought a book cannot read. So he was asked to explain his research in 7 words. So he said that DNA bought a book cannot read. So we actually were invited to a podcast where I said exactly this thing, that it's not about reading a book. It's also about feeling or perceiving the structure of a whole Lego model, if you will. So I think we are totally excited to pursue those directions and bring all of our expertise. And I think we are also learning new machine learning approaches. Like we are borrowing a lot from like cutting edge machine learning techniques to enrich these models, because many of these models, we take ideas from established nucleotide sequence based models, but once we go to the continuous data, DNA shape, their angular measurements, their continuous values, we need new models. So it's like a great experience for us from all perspectives. So probably you will see me banging your door very soon, right after the talk.
Okay. Okay. Yeah. Thank you very much.
Sounds good. So any other question from audience?
Can I ask a question?
Go on.
Hey, Dr. Samee, that's a great talk. So I have a question according to the DNA shape, like the shape feature you got is from the DNA sequence only, right?
That's right.
Okay, yeah, I was wondering like whether there is different cell types or how is the consistency between different cell types according to their DNA shapes. But yeah, if you got it from only DNA sequence, I think that won't be an issue, or it doesn't matter anymore. Or
I think that's a very like legitimate question, and many of the answers that I give to these in response to these questions, I actually learned those answers from Remo Rose himself. So in response to these kind of thoughts, what we all think is these DNA shapes, they're derived from very small nucleotide context, in this case from 5 mar long nucleotide context. So even though the same DNA region, it might stay linear or it might be a part of like a longer coiled region in one cell type versus another cell type,
right?
The exact deviation in that 5 mar context should not be remarkably different. So that's how we have convinced ourselves. And of course, the data from different contexts supported us better. However, we never tried explicitly how well correlated are these DNA shapes to strongly nucleosomal DNA sequences. And that's one thing that we just started to study from a very recent data set from Johns Hopkins University's Taek-Zip Ha's lab. They have a very detailed analysis of East chromosome. They do not do it exactly from DNA shape perspective, like different geometrical measurement perspective. They kind of put everything together in a more phenomenological term. They call it bendability. So they have measured bendability across the entire East chromosome and on thousands of random genomic sequences. I'm sorry, on thousands of random DNA sequences, and also high nucleosome occupancy sequences in the East chromosome. So now this is a better test bed for us to see if our DNA shape features that we are taking from Dr. Remo Rose's library, how well predictive are they of the bendability data? And if not, then Dr. Lynn Zidrick here at Baylor, they're willing to run more DNA shape analysis and build a better model for DNA shape. So I think there are more opportunities to improve on the shape data itself. And we just need to focus on the right type of genomic regions. So right now, I think we are going to look at the highly bendable, the nucleosomal sequences, and learn some ways to improve the shape data, if it is necessary.
Okay, okay, I got it. Thank you so much.
Sure.
Great. So any other question? I have one question for you, Hassan, and I think that you kind of hit upon it when answering Ringyu's question, that these nucleosomal positions and then the 3D structure of the genome, do you think that the shapes in these regulatory region dictate the packaging of the DNA or vice versa? And or it's a combination of these?
Shape dictating. To be honest, I don't think that any one of us knows a definitive answer to that, right? What I have learned by looking at the data and speaking with other experts, I think yes, shape makes certain parts of the genome more permissible or more plastic to coiling around nucleosome, to becoming more susceptible to mutations, for example. So that's another data set we are now, I'm coming back to answering your question very soon, but there's another data set we are trying to gather where there are mouse models put under mutagenic stress. And the question is again, why are certain parts of the genome more mutated under environmental mutagenic stress like UV light and so on? Is there like some structural perspective to explain the data better and so on? So I think to a large extent, yes, the DNA structure makes certain parts of the genome more permissible or more inducible to bend and wrap around nucleosome to make those long distance communications. Yes, because ultimately the long distance communications are mediated by different protein machinery trying to pull them together, which is again bendability at a much bigger scale. So in my naive thoughts, yes, but I also saw these thoughts matching with the experts as well.
Very, very nice. Thank you. And like just a very touch of a answer, kind of like, do you think that such shapes exist for RNA? We know that RNAs, they have secondary structures and a shape in the kind of like how they fold. But do you think that at the base level, the shapes that we see in DNA structure would exist in the RNA structure as well?
That's a fascinating question. I mean, we have thought about that. I think that needs some basic statistical valid. I mean, hypothesis testing at the fundamental level, because I I really don't know. I really don't know. I am biased to say yes, because one way to answer many of these questions that Remo Rose does is when a protein binds to the DNA, it doesn't travel with a dictionary of k-mers. All it cares about is the electron density at the interface of its DNA binding site. Now, bringing that biophysical understanding to this RNA level, I would say that it's that the time scale that it takes for a RNA to fold and eventually to mature to secondary to tertiary and to a complete protein, in that time scale, it's necessary to have some systematic clue from the beginning. So I am biased to say yes, that it is embedded in the structure, in the geometry. And we all know that mutations may disrupt those initial like structures and disrupt ultimately the folding. So
Perfect.
But if there are data set, I'm totally excited to try and come up with at least some p-value for you. I know that you won't buy my words.
Well, I think your words for me is more than p-values. So, so I accept that. No worries. Wonderful, wonderful. Thank you so much, Hassan. That was a fascinating talk and a lot of directions. I learned actually a lot of stuff from just listening to this. We'll be in touch shortly, and I think we are going about 15 minutes after our time because it was very exciting. So let's thank Hassan again for coming and giving us this wonderful talk. Thank you so much, Hassan. And we'll be in touch. Okay?
We will definitely be in touch. Thank you, Dr. Kihara, for inviting me. It was my privilege. Thank you so much.
Thanks,
Thank you.
Hassan.
Thank you for your time. Thank you.
Bye. Thanks. Bye.
It's great touching base with you.
Sure. Thanks. Bye.
All right.
Right.
Bye.
Bye.